When you're trying to determine primary production in any aquatic habitat, whereas in a lentic habitat, the light and dark bottle method works the best. Remember, that is in lakes or ponds. For a lotic habitat, such as this trout stream, the best technique to use is the diurnal curve method. It also involves oxygen levels, changes in oxygen levels in the water over a 24-hour period. It has that in common with the light and dark bottle method, but some of the technique is a little bit different. I think the first thing we ought to do is talk a little bit about oxygen pulses within a stream such as this during a 24-hour period. Now, it's a very different type of habitat, obviously, than a lake or a pond. You have flowing water. You have water that shows a great deal of turbulence, so there's a lot of mixing between the water and the air, and that adds a lot of oxygen to the water. Also, there's a lot of surface area per volume. That is to say, the water, most of the volume is in contact with the air. Obviously, in a lake or a pond, only the very surface is in contact with the air, and other than wave action, there's not a great deal of turbulence. The first thing to determine is saturation in this stream during the 24 hours that you're going to determine primary production. Now, by saturation, I mean the amount of oxygen that water at this temperature would normally hold. Now, let's assume that the temperature is roughly about 10 degrees centigrade or Celsius. Saturation at that temperature is roughly around 12 milligrams per liter. In other words, that's what it would normally hold. So that during that 24-hour period, your baseline would be 12 milligrams per liter of oxygen. Now, in headwaters such as this, where there is a great drop, a steep gradient in terms of the stream, you're going to have a tremendous amount of white water, therefore a lot of turbulence. All of that turbulence means that it would be quite normal during that 24-hour period for the oxygen curve to be above the saturation line. That is to say, instead of 12 milligrams per liter, not involving photosynthesis or respiration of organisms, you might expect that your diurnal curve, that is your 24-hour curve, would be slightly above saturation throughout the 24-hour period. Now, superimposed on top of this is the effects of life in this stream, whether it's photosynthesis, which produces oxygen during the day. Remember, and we talked about it before, photosynthesis liberates oxygen as a byproduct, or respiration, which removes oxygen from the water. Let's take a look at our two major components of our trout stream community. We have the producer component. These are the green plants. These are organisms that photosynthesize during the day, but remember, they respire at night. So whereas they're producing oxygen during the day, they're removing oxygen at night. The other major component of the community, of course, is the animal life, the invertebrates, the fish. They are respiring 24 hours a day. Now, if this were a habitat where there was a lot of organic material, we'd also have to include as a major component our bacteria. That is, the decomposers, which would also respire 24 hours a day and form what we refer to as BOD, or biological oxygen demand. But in a habitat such as this, the bacteria really have a minor effect on our diurnal curve, so we'll work with just the producers and the consumers, that is the plant population and the animal population. Now let's take our curve and plot it on a graph during this 24-hour period. And we're going to plot it on the horizontal axis. At the two ends of our graph will be 12 midnight at the beginning of the period, 12 midnight at the end of the 24-hour period. Halfway through will be 12 noon. In other words, 12 hours from either end. What's our graph going to look like? Well, it depends very much on the habitat we're working in. If we're working in the headwaters, I have to take into account what the community is going to contribute. This is a relatively sterile environment. That is to say, there are very few nutrients at this point. So both your producer population, the plants, and the consumer population, the animals, are relatively low in number. Yeah, there's a lot of diversity, as we've mentioned before, but not an awful lot in terms of total numbers. So your curve, and remember, you're plotting this against or along with the saturation line for 24 hours. Your curve is going to be slightly above saturation. During the dark hours, it'll be very close to or at, maybe even a little below saturation when all the organisms are respiring. In the noon hour period, that is during the light hours, there'll be some photosynthesis. So your oxygen level is going to rise slightly above saturation, but the whole point is the diurnal curve in the headwaters is going to show very little variation, a slight increase during the light hours, a slight increase during the dark hours. Now, let's take that same curve further downstream. Now, downstream, we're going to end up with more nutrients coming in from runoff from neighboring communities, any sewage, road runoff, any field fertilizers, any limestone, sedimentary formations contributing nutrients. There, you're going to have a greater plant community, and therefore a greater animal community. Producers are up, consumers are up. That's going to have a major effect on your oxygen curve. Also, remember that there'll be less turbulence because your gradient is dropped. So that with less turbulence, your line automatically is going to be right about at saturation. You're not going to have a slight elevation above saturation because you're not going to get supersaturation from turbulence. What's your curve going to look like? It'll have the same shape. It'll be basically bell-shaped, as we've talked about before with this area, but it's going to be a much greater magnitude. At night, you're going to have much more respiration, and therefore your curve is going to be well below saturation. During the daytime, with strong photosynthesis, your curve is going to be greatly above saturation. Fine, that's the shape of the curve. How do you determine primary production from those curves? Well, actually, basically the same as the light and dark bottle method. At nighttime, you have total respiration. That is to say, the area below the saturation line, the area formed between the saturation line and your diurnal curve, reflects not only respiration by the consumers, but remember the plants are respiring too at this time. So the areas below the curve at either end, total respiration or R. Now that area above the two curves, that is during the daytime, the line representing your saturation line, and then your diurnal curve, the area between those two is photosynthesis, which is total assimilation, minus respiration. So what you're getting at that point is P sub G minus P sub N, or R. So, like the light and dark bottle method, you have a handle on gross production, net production, and respiration, and of course you can calculate your primary production from this. As I mentioned, those are the two best methods, the most commonly used methods, for determining primary production in a lenic habitat, light and dark bottle, a lotic habitat, the diurnal curve. But there are other techniques. One of them that's used not only in aquatic, but more commonly in terrestrial habitats, is known as the harvest method. The harvest method suggests exactly what's involved. And what it involves is going into an area of a standard size, or a size that you know, whether it's an acre or a half acre or whatever, going in and harvesting or removing all of the production in that area. Now remember, and it's important to remember, that productivity means a certain amount of material produced, biomass, in a certain area, hectares, acres, in a given amount of time, in hours or months or whatever time limit you use. Let's say you're working with a cornfield for the harvest technique or the harvest method. Well, you know the area, the corn's being grown in a given acre. You know the amount of time, that is, that corn grew for 4 months, and you know the biomass because you can go in and harvest that corn and weigh it and know the amount of weight. So you can get an exact indication of productivity because you have weight per area per time. This can be applied to streams as well, working with higher vegetation. That's the harvest method.